Neurological Disorders, Lesson 5.6. What are the long-term effects of drug abuse and addiction? The goals of this lesson are to demonstrate the long-term consequences of drug abuse and addiction, and to describe how drug abuse changes neuronal structure and function. At the end of this lesson, students should be able to describe how drug abuse alters protein expression, describe how changes in protein expression alter synaptic structure and function, and describe the neural correlates of tolerance and withdrawal. We'll achieve these goals through patient diagnosis and Socratic discussion. To prepare for this lesson, you'll need to review the key scientific concepts presented in it. They include the factors to consider before determining if someone is addicted to a drug, the central dogma that DNA codes for RNA, which codes for protein, the mechanism by which long-term drug abuse causes changes in protein synthesis, synaptic structure, and neural circuits, and the neural mechanism behind tolerance and withdrawal. You can review the scientific content in the background reading provided to you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in the lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of lesson structure, including instructions on how to manage the discussion and activity, and the student workbook provides additional explanations for your students. The key point of the lessons due now is that motivation is a key factor to consider when determining whether someone is or will become addicted to a drug. We'll get to this key point by reviewing last night's homework. In the Do Now, students work with a partner to review last night's homework about drug use by soldiers during the Vietnam War. They'll focus on two specific questions: what contributed to drug use in Vietnam, and what happened when the soldiers returned home, as both of these are central issues to consider when investigating addiction, and the answers to these questions are why the case study of drug use in Vietnam is a classic case study when studying addiction. The key points of the discussion are the factors to consider before determining if someone is addicted to a drug, its central dogma, that DNA codes for RNA, which codes for protein, that long-term drug abuse causes changes in protein synthesis, synaptic structure, and neural circuits, and changes in neural circuits underlie tolerance and withdrawal. We'll get to these key points with a Socratic discussion. The discussion starts with a brief story of two people, Pat and Chris, who have both been using morphine. The cases of these two people allow the class to discuss important factors to consider before determining if someone is addicted to a drug. Because at first presentation, it appears that Chris is addicted because he is taking more of the drug. But in fact, with later information, you learn that Chris is taking the drug for medical condition, whereas Pat is taking it to relieve psychological stress. And her drug use is compulsive despite adverse consequences. So in fact, Pat is the one addicted. Again, these case studies allow for further discussion of the factors that are important to consider when studying addiction, not just the dose of the drug, but also someone's motivation or reason to take it. The discussion then transitions into what's happening biologically in the brain during addiction. First, by comparing the structure of two neurons, a control and one from Pat's brain. The addicted brain has many more synapses than the control brain. And then going into the biological mechanisms that explain the massive increase in synapses following addiction, by asking students leading questions to build the central dogma. First, by asking, what caused these changes? What are neurons composed of? Proteins. And then discussing how are proteins changed. But first, what within the cell contains the code for proteins? DNA. Where is DNA stored? The nucleus. Where are proteins synthesized? The cytoplasm. How does the DNA from the nucleus get into the cytoplasm for proteins to be made? RNA transports the code to the cytoplasm. And then by reviewing the central dogma, that DNA codes for RNA, which codes for proteins. The next section of the discussion ties the central dogma and change in DNA expression to changes in synaptic structure, by first showing students that neuronal stimulation causes protein synthesis, and overstimulation increases protein synthesis, and finally, overstimulation increases the number of synapses. Slides 16 and 17 tie the addition of synapses to the brain's reward circuit to allow discussion of the mechanisms behind tolerance and withdrawal. After reviewing the reward circuit with your students, you'll ask them, once abnormal synapses have formed, would you need more or less of a drug to activate these synapses? You would need more, and that underlies the phenomenon of tolerance, needing more of a drug to have the same biological effect. After presenting the definition of tolerance to your students, you'll ask them, can you think of an example of tolerance in your daily life? The picture of the coffee cup usually does the trick, as many high school students have developed a tolerance to caffeine. You'll then ask the question, once abnormal synapses have formed, what would happen if you stopped using a drug? The network would function abnormally, and you'd experience withdrawal. Withdrawal is the wide range of symptoms that occur after stopping or reducing the amount of drug consumed after heavy or prolonged use. After presenting the definition of withdrawal to your students, you'll ask them, can you think of examples of withdrawal in your daily life? Again, the picture of the coffee cup usually does the trick, and usually there are several students who've experienced a morning without coffee and can describe the headaches, difficulty focusing, and extreme cases, feeling of sickness. The key points of the wrap-up: the DNA expression and structures of the reward pathway is altered by repeated drug use, and that drug use causes an increase in the number of synapses in all the structures of the reward pathway. We'll get to this key point with a reflection and discussion. In the wrap-up, we'll again review the reward pathway is composed of the VTA, nucleus accumbens, and prefrontal cortex, and address two questions. First, the DNA expression of which of these structures is altered by drug abuse, all of them, and how does this alter their physical structure? Drug use increases the number of the synapses. The key point of the homework is that there are a variety of treatment strategies for drug addiction. We'll get to this key point by researching treatment strategies for drug addiction. Students often ask for more details about the changes in our brains as a result of addiction. You can find more in the Teacher Primer, Teacher Manual, and Student Workbook. One of the most common questions teachers get is how long after someone stops using drugs do the changes in their brain remain? That can really depend on a variety of things, including the drug the person used, the length of time of use, and the age at which the drug addiction started, to name just a few. But evidence is indicating that the changes in the brain last long after drug use stops, that synaptic connections created are stable and may underlie relapse. At the end of this lesson, students should appreciate the variety of factors that are important to consider before determining if someone is addicted to a drug. Additionally, students should appreciate the long-term changes that occur in brain structure and function, as well as the biological mechanisms behind those changes. This lesson introduces the idea of addiction as a chronic condition resulting in long-term changes in our brains, an idea we'll develop more fully in the next lesson. This lesson also provides a foundation for future discussions of addiction as a disease. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members, and we'd be more than happy to help you.